Hi everyone, I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with cholesterol pool and cholesterol metabolism. In today's session, we will study excretion of cholesterol. Then we have a classification of lipoproteins and apolipoproteins. And lastly, we have metabolism and functions of chylomicrons. Good morning, everyone. Let us begin the topic. Excretion of cholesterol. Average diet contains about 300 mg of cholesterol per day. Body synthesizes about 700 mg of cholesterol per day. Out of these 1000 mg, about 500 mg of cholesterol is excreted through the bile. This cholesterol is partly reabsorbed from intestine. Vegetables contain plant sterols, which inhibit the reabsorption of cholesterol. The unabsorbed portion of is acted upon by intestinal bacteria to form cholesterol and prostanol. These are excreted through intestines. Another 500 mg of cholesterol is converted into bile acids and which are excreted in bile as bile salts. Coming to liver and cholesterol. The liver has a major role in controlling the plasma levels of LDL cholesterol. Liver synthesizes cholesterol. Liver removes the cholesterol from lipoprotein remnants. Liver is the only organ that can excrete cholesterol through bile, and liver converts cholesterol to bile acids. Coming to plasma lipids. Total plasma lipid is 400 to 600 milligram per day deciliter. Out of this, 40% is cholesterol, 30% is phospholipids, and 20% is triacylglycerides. Since lipids are insoluble in water, they need the help of carriers in plasma. Therefore, they are complexed with proteins to form lipoproteins. The protein part of lipoprotein is called apolipoprotein. The lipoproteins are usually abbreviated as LP. Coming to classification of lipoproteins. Depending on the density by ultracentrifugation or electrophoretic mobility, the lipoproteins in plasma are classified into five major types. First, chylomicrons. It contains apoprotein B48. Second, very low density lipoproteins or pre-beta lipoproteins. Main apoprotein is B100. In this diagram, we can see that depending upon the density, the the lipoproteins are classified by ultracentrifugation or electrophoresis. In this diagram, we can see that lipoproteins are diminishing in size from chylomicron to HDL. Coming intermediate density lipoproteins or broad beta lipoproteins. Then fourth is low density lipoproteins or beta lipoproteins. Major apoprotein in LDL is B100. Fifth is high density lipoproteins or alpha lipoproteins. Major apoprotein in LDL is ApoA. Free fatty acids and non-esterified fatty acids are complexed with albumin. FFAs, that is free fatty acids, are not included in the classification of the lipoproteins because they are loosely bound to the protein. Coming to general characteristics of lipoproteins. The lipoproteins molecules have a polar periphery made up of proteins and polar heads of phospholipids and cholesterol. The inner core consists of hydrophobic triacylglycerols and the tails of phospholipids. The apoproteins also increase the solubility of lipids. Coming to separation by ultracentrifugation. The lipoproteins are characterized on the basis of their density. Fat is less dense than water, so fat floats on water. Lipoprotein with high lipid content will have a low density and so they float on centrifugation. Those with a high protein content will sediment easily and have a high density. Depending on the flotation constant, the different fractions can be separated. So, coming to separation by electrophoresis. The serum is applied on cellulose acetate. Uh, Electric current is applied for 2 hours and the strip is tied and strained with lipid dyes such as oil red O. As a general rule, those with higher protein content will move faster towards the anode. Few proteins have minimum mobility. Coming to apolipoproteins. The apoprotein part of lipoprotein is called apolipoprotein or apoprotein. All apoproteins are mainly synthesized in liver, but small quantities are produced from all organs. Intestinal cells produce small quantities of ApoA. Apart from solubilizing the lipid part, the protein components have specific functions. Number one, ApoA1. It activates the lecithin cholesterol acyl transferase. It is the ligand for HDL receptor. It is anti-atherogenic. Number 2, ApoB100. It is a component of LDL. It binds to LDL receptor on tissues. ApoB100 is one of the biggest proteins, having 4,536 amino acids with a molecular weight of 550 kilodaltons. It is synthesized in the liver. Here we can see the fatty acid pathway for the uh, intestine. First, it enters into the intestine, then into nascent chylomicrons as triacylglycerols and cholesterol. Then it enters the mature chylomicrons as triacylglycerol plus cholesterol. Then it enters into the muscle and adipose tissue. Then in the muscle tissue, it is converted into chylomicron remnants, mainly cholesterol. Then it enters the liver, mainly ApoE receptors. Third is ApoB48. It is synthesized in intestinal cells. It is the structural component of chylomicrons. ApoB100 and ApoB48 are products of the same gene, but in the intestine, the mRNA undergoes editing so as to produce the B48 protein. B48 is so named because it is only 48% of the size of B100. Here we can see ApoB100 mRNA. On the left side, it enters into the intestine where there is a stop codon in between, producing truncated protein that is ApoB48. On the other hand, in the liver, there is no stop codon, so protein is fully translated, which is called ApoB100. Number 4 is ApoC2. It activates lipoprotein lipase. Number 5 is ApoE. It is a arginine rich protein. It is present in chylomicrons, LDL and VLDL. Astrocytes also make up ApoE. It is involved in cellular transport of lipids in CNS. ApoE has 1, 2, 3 and 4 isoforms due to independent alleles in the gene. ApoE4 isoform is implicated in the development of senile dementia, Alzheimer's disease, and lipoprotein glomerulopathy. Coming to synthesis of chylomicrons. Chylomicrons are formed in the intestinal mucosal cells, and secreted into the lacteals of lymphatic system. They are rich in triglycerides. If lipemic serum is kept overnight in the refrigerator, chylomicrons rise as a creamy layer to the top, leaving the subnatant clear. Here is a diagram of the chylomicron, which is which contains cholesterol, ApoB48, ApoA, ApoC2, phospholipid outer cover, and triacyl triglycerides in the interior. When the chylomicrons are synthesized by the intestinal mucosa, they contain only ApoB48 and ApoA, but ApoC and ApoE are added from HDL in blood during transport. Coming to metabolism of chylomicrons. Main sites of metabolism of chylomicrons are adipose tissue and skeletal muscle. The half-life of chylomicrons is about in blood is about 1 hour. The enzyme lipoprotein lipase LPL is located at the endothelial layer of capillaries of tissue at muscles and heart, but not in liver. ApoC2 present in the chylomicrons activates the LPL. The lipoprotein lipase hydrolyzes HDL, hydrolyzes triglycerides present in chylomicrons into fatty acids and glycerol. Muscle or adipose tissue take up the liberated fatty acids. Following injection of heparin, the LPL is released from the tissues and lipemia is thus cleared. This is called post-heparin lipolytic activity. Lack of CpC2 leads to decreased activity of LPL and consequent accumulation of chylomicrons and VLDL in blood. B. Insulin increase the LPL activity. Coming to liver chylomicron remnants. As the triacylglycerol content is progressively decreased, the chylomicrons shrink in size. These remain containing ApoB48 and ApoE are taken up by hepatic cells by receptor-mediated endocytosis. ApoE binds the hepatic receptors. Coming to functions of cholesterol, chylomicrons. Chylomicrons are the transport form of the dietary triglycerides from intestine and to sub adipose tissue for storage and muscle or heart for their energy needs. Here ends the session. Kindly like, share and subscribe the channel. Thank you.